random amplified polymorphic DNA and amplified fragment length polymorphism. These are the two techniques which are also called as molecular markers. In case of random amplified polymorphic DNA, that is a PCR based marker that consists of 10 to 12 base pair longs. So these markers are used to perform the random polymorphic DNA sequences. Random amplification of several fragments with the help of these 10 to 12 base pair long markers, and the amplified fragments, they are run on agarose gel detected by the ethidium bromide. Unstable amplification leads to poor repeatability. So in case of random amplified polymorphic DNA, which is also called as rapid, in case of rapid, there are 10 to 12 base pair long markers. With the help of these markers, DNA is amplified and it amplifies several fragments of the DNA. So amplified fragments, they are run on the agarose gel and they are detected with the help of ethidium bromide. So this technique is called as random amplified polymorphic DNA. While on the other hand, amplified fragment length polymorphism, in this technique, restriction endonuclease are used for the digestion of the DNA. Once restriction endonuclease enzymes are used for the digestion of the DNA, then there is ligation of the adapters. Adapters are attached on the ends of the DNA. Once adapters they are attached, then there is amplification of the ligated fragments, mean those fragments that have the DNA and on both sides of the DNA there are adapters. Once they are amplified, then those ligated fragments or the amplified products, they are separated with the help of gel electrophoresis and they are visualized. So AFLP have stable amplification and good repeatability as compared to the rapid. So these are two other techniques which are also used as a molecular markers, which is due to the polymorphic DNA sequences. If we do the comparison between all these techniques like RFLP, restriction fragment length polymorphism, RAPD, random amplified polymorphic DNA, STR, short tandem repeat, SNP, single nucleotide polymorphism. If we do the comparison between all these techniques based on the detection method, in case of detection method, here we use hybridization. In case of these three techniques, we use polymerase chain reaction. Based on the type of the probe or primer used, here genomic DNA sequence is used or cDNA sequence is used in case of RFLP. While in case of rapid, arbitrarily a primer is designed to perform the rapid RAPD. In case of STR and SNPs, these are due to the sequence specific primers. Now coming towards the requirement of the radioactivity, in case of RFLP, that is yes, there is a need of radioactive labeled probes, while in case of these three techniques, it depends whether there is a need of radioactive element or not. But in case of now STRs and SNPs, most of the fluorescent dyes are used to perform the STR analysis or to perform the SNP analysis. Degree of polymorphism, in case of RFLP it is low, while in case of rapid it is medium to high, and these two techniques STR and SNPs, they have high degree of polymorphism. So molecular markers are different techniques to study the mutations in the DNA. These techniques are also used to study the coital traits which are present in the plants or in the animals.